Good afternoon, evening. I'm excited to share with you some of my lab's research and describing your methods and applications of isotope tracing and mass spectrometry to probe cancer metabolism. So to start, I'd like to point out one of the problems that we need to overcome in studying and understanding metabolism, and that the cells in our body, we have a different set of cells that perform different functions, be they neurons, muscle cells, or cancer cells. And throughout our lives, they're exposed to a number of different stresses, drugs, microenvironments, and nutrient conditions, which ultimately will combine to generate a wide array of metabolic phenotypes. And all of these can be represented by changes in functional metabolic networks. So a critical challenge facing us is to characterize how metabolism is perturbed in these different situations. So this is particularly true in the case of cancer, where we know that tumor cells exhibit very different metabolism than surrounding normal tissues. So as I noted, aerobic glycolysis, which is exhibited by virtually most cancers and proliferating cells for that matter as well. And this is indicated by increased glucose uptake. And we commonly exploit this for diagnostic purposes in the form of FDG PET imaging, and a picture of that is shown here. We use this as a great tool to study sites, to identify sites of metastasis or tumor sites in patients. But this only scratches the surface with respect to cancer metabolism. So a question that we want to put forth is, can we study cancer metabolism more deeply to identify and exploit specific metabolic dependencies in tumor types? So today I'm going to describe to you some work in which we've focused on IDH mutant tumors to identify specific metabolic dependencies, and then describe some more recent technology development to look at different metabolic pathways in cancer cells. In any talk like this, I always find it very useful to give an overview of central carbon metabolism or intermediary metabolism to refresh myself and the audience with these pathways, because we're going to be talking about it in good detail today. But so in glycolysis, glucose is lysed into 2 molecules of pyruvate. And what Warburg demonstrated is that cancer cells exhibit aerobic glycolysis, so they selectively divert much of this pyruvate to lactic acid. And we think this occurs, but we don't really know for sure why, but we think this occurs to facilitate biosynthetic flux of intermediates toward nucleotides or lipid biosynthesis. Now, pyruvate can also be transported into the mitochondria and oxidized to acetyl-CoA, where it will enter the TCA or Krebs cycle within mitochondria. And these pathways are coupled to cellular bioenergetics and respiration to generate ATP, and also used in both mitochondrial and cytosolic reactions to generate biosynthetic intermediates. Now, we've learned over the last 2 decades that these metabolic pathways are regulated by a number of different oncogenes and tumor suppressors. Some of those are shown here in blue. We've also seen, and you'll see more of this here, that the microenvironment itself will exhibit profound effects on how cells metabolize their surrounding nutrients. And even more recently, we've learned that metabolic enzymes themselves are differentially expressed or commonly mutated in specific types of cancer. And some of these specific enzymes are listed here. So the challenge for us in understanding cancer metabolism is to translate the metabolic pathways that I've just, two of which I've just described to you, and they're depicted in this map on the left here, into functional readouts of metabolism or metabolic fluxes. Because the molar conversion rate per time or per protein or cell, or the metabolic flux, represents the ultimate metric describing enzyme metabolic function. Now, over the years, the last 30 years or so, we've developed new technologies which allow us to better probe metabolite abundances in biological tissues. These range from mass spectrometry to NMR. They're highly quantitative. Often you can discover metabolite changes in a non-targeted manner, making them very attractive discovery tools. But they can be of somewhat limited utility in describing metabolic fluxes. So shown here, if we think about a simple reaction scheme, two reactions catalyzed by enzyme X and enzyme Y, if we analyze metabolite levels and see that metabolite C becomes increased in the context of a given disease, we have no way of knowing if this is because of increased activity of enzyme X or decreased activity of enzyme Y. So, and if we extrapolate this to a larger map where there may be 50 or 100 enzymes that will metabolize metabolite C, you can see that this gets pretty complex. So the way we circumvent this issue is by incorporating the use of isotope tracers and applying some systems-based analyses of our data sets. And that's the general approach that we take in the lab. We apply metabolic tracers to a number of different mammalian cell systems, including cancer cells, animals, pluripotent stem cells, a combination of analytical chemistry tools, including mass spectrometry-based metabolomics, and then some systems-based modeling to characterize and quantify intracellular metabolic fluxes. And we apply this to a number of different systems in the lab. Today I'll share with you our work on IDH mutant tumor cells and some new methods that we've developed to probe cellular redox states in specific cellular compartments. So just one more quick, simple example to describe the utility of these isotope tracers. In this toy example shown here, I have a background in chemical engineering, so you can think of this as a set of different reactors that have liquid flowing from one tank to the next. This is a common problem in engineering, and one way to solve that is to incorporate the use of an isotope tracer. So for example, we may have clear fluid running into one side, and what we can do is put a dye in to another input, and this will generate a distribution of color in the different reactor sets. Now that color distribution is a direct function of the flow from one reactor to the next. Now metabolism is no different, but instead of a set of interconnected reactors or tanks, we have the Krebs cycle shown here, where you have glucose and glutamine carbons flowing into the TCA cycle. But rather than put a dye, what we'll do is incorporate stable isotope labeled tracers. So depicted here, and you'll see these type of maps throughout the seminar. These circles represent carbon atoms that are either unlabeled or carbon-12 labeled or isotope labeled and 13C labeled. And as labeled glutamine enters the Krebs cycle, this mixes with unlabeled carbons from glucose and pyruvate to generate a distribution of isotopomers, of mass isotopomers. So what we can then do is extract metabolites from this cultured cell system and analyze isotopic labeling in citrate, for example, via mass spectrometry, and ultimately use that and incorporate that into some models, assuming steady state or in non-steady state conditions, accounting for metabolite abundances to get at the metabolic fluxes in our system. So one way we commonly do this is to address how cancer cells or other cell types generate fatty acids and lipids. And the key metabolite here that we're interested in is acetyl-CoA, which gives rise to fatty acids when metabolized by fatty acid synthase. So shown here, we have isotopically labeled glucose, which is converted to labeled acetyl-CoA. And if we culture cells for a period of time, we can see that labeling is incorporated into newly synthesized fatty acids. So shown here is a quantitation of the number of isotopes per molecule, or the mass isotoper distribution on newly synthesized lipids. And what we can see, get from this information with some downstream labeling, is both how many fatty acids were newly synthesized, and by knowing the number of isotopes per molecule, we can determine the contribution of our tracer, glucose in this case, to the lipogenic acetyl-CoA pool. And in this case, we can see that about 80% of lipid carbon comes from carbohydrates, or glucose in this case. Now, this isn't always the case. We can also, glutamine is also utilized for fatty acid synthesis, as shown here by a 513C labeled glutamine, which is incorporated into lipogenic acetyl-CoA, albeit at a lower level. But one interesting thing we saw is that when we place cells under hypoxia, this pattern switched, and we saw a very significant enrichment of newly synthesized fatty acids of our isotopically labeled glutamine carbons into newly synthesized fatty acids, such that under hypoxia, the majority of the lipid carbon came from glutamine, which was actually metabolized in this reductive pathway, such that it's metabolized in the reverse direction of the Krebs cycle, as is normally thought to be the case. And we can back out these contributions to the acetyl-CoA pools with some simple modeling, and we and others have recently shown that this is pretty common to a number of different cell types. Under hypoxia, glucose is minimally utilized for lipogenesis under hypoxic conditions, whereas glutamine, metabolized through this reductive pathway, is the preferential carbon source used for this process. Now, we've looked at how this occurs in part through hypoxic induction of HIF and PDKs downstream. And what we found is that normally, if we take a compound dichloroacetate, which inhibits these PDK1, which in turn inhibits pyruvate dehydrogenase flux, it really has a minimal effect under normoxic conditions. Under hypoxia, we see that this glutamine pathway is highly upregulated. But if we treat cells with DCA, which in animal models exhibits anti-tumor activity, we blunt this switch to glutamine to the use of glutamine for lipid synthesis and force cells to use more glucose, as shown here. This pathway is particularly important in some cancer cells, as we've seen, and in particular, some tumors from patients with von Hippel Lindau syndrome, such that they lose this VHL tumor suppressor protein and have constitutively active HIF And surprisingly, we found that these cells are always dependent upon glutamine for their lipid synthesis. And VHL deficient tumor cells, even under normoxic conditions, preferentially use this glutamine pathway. Whereas kidney cancer cells that have wild-type VHL behave as normal cells do and switch under hypoxia. This isn't completely specific to tumor cells, but we also see some normal tissue types use this, such as T cells and adipocytes in our own lab now. Another example of a system where mitochondrial substrate utilization, demonstrating how flexible it is some recent work we've conducted in our own lab, where we've exploited this newly identified mitochondrial pyruvate carrier, which is the transporter through which pyruvate enters the mitochondria. So this was the genes encoding this transporter were identified just 2 years ago, and what we've done is observed how mitochondrial substrate utilization changes when we knock down MBC1 or MBC2 or treat cells with an inhibitor of this mitochondrial pyruvate transporter. And this manuscript is actually just published online today in Molecular Cell, along with some other manuscripts which describe similar effects in cancer and muscle cells. And if we knock down MBC1 or MBC2, it actually eliminates expression of the other gene products. But surprisingly, we saw little effect on respiration. And only if we added an uncoupler to the cells did we see a change or an effect on respiration and a reduced respiration. But basal respiration was constant. And cell growth itself in cancer cells or myoblasts also remained constant. And we observed no change in glycolysis or lactate efflux. What we did see is a profound change in amino acid metabolism, and I'll just show you the results with glutamine here before moving on to the IDH cancer studies. But what we see is that the contribution of glutamine versus glucose to lipogenic acetyl-CoA switched almost completely, such that the cells preferentially utilize glutamine for lipid biosynthesis, just like was the case for hypoxia. However, in this situation, a different pathway is used, and we see this oxidative glutaminolysis pathway that is selectively used when we knock down the mitochondrial pyruvate carrier. Now, this gives an indication of how flexible some cell metabolism and intermediary metabolism are. But how do these pathways impact specific cancers and mitochondrial physiology in general? So some of the questions we've looked to address is how these pathways are modified and affected in cells expressing a mutant IDH1, and what is the role of cofactors such as NADPH in these processes. So if I rewind to this map of intermediary metabolism, I'll highlight the role of IDHs in this, and particularly their discovery and involvement in cancer. So in about 2008, it was first discovered in some exome sequencing of gliomas that there were recurrent mutations in IDH1, and then subsequently it was seen that IDH2 was also mutated in a subset of gliomas, as well as some AML tumors and some other sarcomas as well. Subsequent work demonstrated that these mutant enzymes, which were always at the same residue and in the heterozygous state, exhibited gain-of-function activity and reductively metabolized alpha-ketoglutarate to 2-hydroxyglutarate. So instead of exchanging alpha-ketoglutarate and CO2 with isocitrate, 2-HG was produced and accumulates at very high levels in these cells. So we wanted to ask, how does this impact cancer metabolism? Now, a lot of work has recently been done describing that this 2-HG molecule influences the activity of a number of different alpha-ketoglutarate-dependent dehydrogenases, and there are about 50 of these expressed in mammalian cells, and they execute a number of different functions ranging from hypoxia-inducible factor stabilization to histone and DNA demethylation. So the specific effects may be a function of different tumors or different tumor subtypes as well. So we've investigated whether metabolism might be a more consistent defect that can be targeted or targeted in tandem with the demethylase or prolyl hydroxylase activity that becomes dysfunctional. So to do this, we've employed an isogenic pair of cell lines in collaboration with some folks at Novartis, and tried to address two questions. How do heterozygous IDH1 mutations affect metabolic flux in these cells, and do they create liabilities that can be exploited? So using a background cell type of HCT116 cells, we studied several clones, and I'll skip over some of the early results, but one thing we found is that under normal growth conditions, we saw minimal effects on metabolism. However, when we grew these cells under hypoxia, we saw more profound changes, and this is consistent with the role of IDH1 and IDH2 in catalyzing that reductive carboxylation reaction in the context of electron transport chain inhibition or hypoxia, as I just described earlier. And in fact, that's what we see when we place these cells under hypoxia. We see profound changes. Shown here are the results of a tracer analysis and model-based output of intracellular metabolic fluxes. Some of the key changes we observed: the IDH mutant cells had a 10-fold reduction in activation of the reductive carboxylation pathway. They also exhibited increased glutamine anaplerosis and oxidative glutaminolysis flux compared to cells with wild-type IDH1, indicating that they were more reliant on glutamine metabolism to metabolize in the oxidative direction of the TCA cycle. Consistent with these changes, in collaboration with Seahorse Biosciences, we've analyzed the respiration of these cells under hypoxia, and what we saw is that IDH mutant tumor cells exhibited higher respiration rates compared to those with wild-type cells when placed under hypoxia. And we saw no change in hypoxia-inducible factor stabilization, so that we thought this was due to metabolic reprogramming and defects under hypoxia resulting from the IDH mutation. So, given the fact that these cells seem to be more dependent upon respiration under hypoxia, we hypothesized that they might be more dependent upon, or they may be more sensitive to inhibition of the electron transport chain. So shown here is treatments with varying concentrations of rotenone, And what we see is that at least 3 different clones expressing different mutations in 2 different mutations in IDH1 all exhibit increased sensitivity to complex 1 inhibition. We saw similar results using independent classes of mitochondrial inhibitors, including fenformin. Interestingly, we also wanted to address whether inhibition of mutant activity was able to rescue this phenotype. So we used an available IDH1 mutant inhibitor and saw a pronounced decrease in 2-HG levels, but saw that this failed to rescue the ability of these cells to catalyze the reductive carboxylation reaction. So we saw no rescue or no increase which we would have expected with the changes in the direction of this red arrow. Here, if the limitation of 2-HG production were to rescue this. So this gives us some sense that inhibiting 2-HG production may be an effective combination approach to also targeting the metabolic defects of these cells, because the mutant inhibitors, which are currently in the clinic, are not rescuing these metabolic defects. So the next question we wanted to address, do IDH2 mutations phenocopy this change? So in this case, we turn to our isogenic panel of H2116 lines containing IDH1 or IDH2 mutations, or alternatively two sarcoma cell lines that express either IDH1 mutations or IDH2 mutations in the case of SW1353 cells. And here I'm using a readout of glutamine conversion to lipids. And what we found is, as before, the IDH1 mutant cells were unable to use this pathway under hypoxia. However, IDH2 mutant cells seem to have no problem and behave similarly to the cells with wild type or cells treated with an exogenous 2-HG. And we see similar trends in comparing the IDH1 mutant HT1080s to the IDH2 mutant cells, which robustly activated this reductive carboxylation pathway, indicating that IDH1 and IDH2 cells differed in their metabolic phenotype. And we also observe this with respect to sensitivity to mitochondrial inhibition. So shown here, treatment with varying concentrations of fenformin, we see that the IDH1 mutant cells are much more sensitive in terms of growth inhibition to fenformin treatment compared to IDH2 mutant cells, which are minimally affected or only affected at much higher concentrations. So, and this work was published earlier this year in Cancer Research. So one of the questions that that came out of this is, what's driving this discrepancy between IDH1 and IDH2 mutations? And this brings forth a general issue in our analysis and investigations of cell metabolism, and that's cellular compartmentalization. So in addition to these central carbon metabolic pathways that I described before, pyridine nucleotides such as NADH and NADPH play an important role in carrying electrons orthogonally throughout the cell and help transfer energy across different pathways and between the cytosol and the mitochondria, though these pyridine nucleotides themselves are unable to be transported into the matrix. So it has to be coupled with intermediary metabolic pathways. So don't be too afraid of these molecules. They're fairly simple. There's a nicotinamide ring, an adenine nucleobase, which are bound to two ribose rings. So nicotinamide, adenine, dinucleotide. And they play an important role in carrying electrons here in the oxidized state, or here there's 2 hydrogen atoms when they're in the reduced state. Now, there's a typo here, but pyridine nucleotides are compartmentalized, and they don't cross over into the matrix. And one issue we have is that 13C tracing is unable to resolve compartment-specific fluxes. And when we normally look at metabolism, we extract cells and get no information about the mitochondrial or cytosolic pools of citrate, alpha-ketoglutarate, or other intermediates within the cell. So we lose all this information in our normal ways of analyzing cells. Yet the cell behaves this compartmentalization is very critical for cell function. So one of the ideas that we've developed recently is to, rather than trace the carbon atom transitions, can we trace the specific hydrogen atoms that get transferred to NADPH and NADH and remain in distinct compartments in this way? And in fact, this works fairly well. So in collaboration with Matt Vanderheiden and Caroline Lewis at MIT, we've developed some methods to study NADPH metabolism in the mitochondria and cytosol specifically. So one of the critical pathways for producing NADPH in cells is the oxidative pentose phosphate pathway. Glucose can be metabolized through this pathway, and this hydrogen atom on the 3 carbon of glucose is transferred to NADPH via 6-phosphogluconate dehydrogenase, the second enzyme in the oxidative pentose phosphate pathway. And this very rapidly labels up the NADPH pool in cells to the point where we get steady state labeling after about 30 minutes, highlighting the fast turnover rate of these metabolic pools. And the NADPH is used for both buffering against redox stress, but also biosynthesis. And in fact, we can see this fairly well. We see NADPH then in turn transferring electrons to fatty acids during synthesis. And just like we saw before with the carbon labeling, we see robust labeling appear on newly synthesized fatty acids, shown here on top, and cholesterol on the bottom. So in this way, we can quantify the contribution of the pentose phosphate pathway to lipogenic NADPH pools. Rather than label the 3 hydrogen on glucose, we can also label the 4 hydrogen, which transfers is transferred to NADH rather than NADPH in the midst of glycolysis. NADH needs to be regenerated through a number of different pathways, including lactate dehydrogenase, the glycerol 3-phosphate shuttle, and the malate aspartate shuttle as well. And after labeling NADH pools, we see robust labeling, not in lower glycolysis, but in lactate, glycerol 3-phosphate, and malate, where this can enter the mitochondrial pool. We see robust labeling over time, which allows us to now look at metabolic flux through these shuttles. So we next wanted to ask whether this we can exploit some of the recently discovered IDH mutations to look at NADPH metabolism in particular in the mitochondrial compartment of cells. So as I alluded to before, as I described before, these mutant IDH1 and 2 convert alpha-ketoglutarate to 2-HG in the cytosol and mitochondria, respectively. So what we did was inducibly express mutant IDH1 or mutant IDH2 in each compartment and detected that the selective expression in mutant IDH1 expressing cells of IDH in the cytosol or the mitochondria of IDH2 mutant cells. And upon DOX addition, we see induced expression of 2-HG. And one of the key points here is this 2-HG acquires label from the mitochondrial or cytosolic NADPH pool. So it can be used as a readout of the isotopic labeling of mitochondrial or cytosolic NADPH downstream of a particular tracer. And in fact, this works fairly well. If we trace NADPH metabolism in the cytosol using the pentose-phosphate pathway tracer described earlier, we only see label transferred to 2-HG in our IDH1 expressing cells that are a readout, that give a readout of cytosolic NADPH metabolism. However, the NADH trace, which enters the mitochondria through the malate aspartate shuttle, we see this label appear in both the cytosolic reporter cells or the mitochondrial reporter cells. So to more functionally test this system, we wanted to determine whether or not we can detect the directionality of compartment-specific reactions in other pathways. So another pathway that's become of great importance, that's garnered a lot of interest in the cancer metabolism community, is folate-mediated one-carbon metabolism, whereby serine and glycine are interconverted and generate one-carbon intermediates, such as 5,10-methylene tetrahydrofolate and 5,10-methylenyl tetrahydrofolate. Now, importantly, these serine hydroxymethyltransferase enzymes and methylene tetrahydrofolate dehydrogenase enzymes are present in both the cytosol and the mitochondrial compartments of cells. And we know that we've known for some time that there's a fair degree of activity in these pathways in all cancer cells that we've tested. So we wanted to ask, if we couple tracing of serine metabolism with our reporter systems, can we determine where this pathway is most active in cancer cells? Is it generating NADPH and NADH in the mitochondria or the cytosol? And we can determine this by using our cytosolic reporter cells and mitochondrial reporter cells. And what we observed is a pretty clear answer, in that we only saw labeling of 2-HG produced by our IDH2 mutant cells. So if we fed deuterated serine to a couple different cancer cells, we only detected labeling in the mitochondrial NADPH pool, but not the cytosolic pool in our cytosolic reporter system, indicating that these cytosolic reactions actually consume NADPH and produce serine rather than be involved in serine catabolism. So if we turn back now to changes in NADPH metabolism as a function of the microenvironment or different disease states, and apply this method, we see additional changes that are occurring in cells if, for example, we put them under hypoxia. So hypoxic cells exhibit increased contribution of the pentose phosphate pathway to lipogenic NADPH, and this is indicated by the hypoxia cells having a shift in this labeling of newly synthesized fatty acids. So this rightward shift indicates that hypoxic cells increase their reliance on PPP flux for producing NADPH. And if we look at this in our IDH mutant tumor panel, we see the same response, where the parental cells and IDH2 mutant cells really increase their reliance on PPP flux to generate NADPH under hypoxia. Consistent with our results before, IDH1 mutant cells are somewhat compromised under hypoxia, and they always exhibit an increased dependence on PPP flux and can't really increase this much more. So this is now leading us to another potential target to be exploited in IDH1 mutant tumor cells, as they may be unable to generate NADPH via IDH1 due to the mutation in that pathway. So to quickly summarize what I've described to you today, our analysis of carbon metabolism and NADPH metabolism in IDH1 mutant tumor cells is identifying some potential therapeutic targets that may be exploited in combination with or independent of mutant IDH inhibitors. Furthermore, more broadly, this combination of C13 and deuterium-based metabolic flux analysis can provide new insights into compartment-specific metabolic pathways and the role of metabolism in diseases such as cancer and others as well. So this latter deuterium tracing work was recently published in Molecular Cell a couple months ago as well. So with that, I'd like to thank you guys all for spending time with me here in this webinar, and thank members of my lab and the wide range of collaborators throughout the country who we work with, and thanks to our funding and thanks to the organizers for having me participate here. So at this point, I'd be happy to, I'll look into addressing some of the questions, if any, at this stage. Okay, thanks all for sticking around to on this late Thursday night, and have a happy Halloween.